We are now going to look at a third category or family of biomolecules that you will discuss at great extent next year in biology. These are proteins, and proteins are polymers made up of amino acids. Remember, polymers are large molecules that are made up of smaller units that are repeated over and over and over again. Like when we talked about polysaccharides, which were polymers of monosaccharides, like starch being a polymer of glucose. In this situation, these amino acids are not exactly the same unit, but they are very, very close to each other. So when you link a bunch of amino acids together, you form a protein. There are several really important functions that proteins perform in the body, and we're going to discuss each of these as we move through this presentation. But first of all, let me just list a few of these. First of all, they make up much of what you are made of, your actual structure. So a great example would be muscle tissue. Muscle tissue is made up of a couple of different proteins that have very special characteristics that allow them to make you mobile, make parts of your body mobile. So structures. A second big function is transportation. I'm not referring to transportation of your entire body. I'm talking about transporting things within your body. So transportation of molecules that need to move from one place to another. Often the conduit are your blood vessels through your blood. A third function is the formation of enzymes or catalysts. Catalysts are any kind of molecule that speeds up a chemical reaction. Enzymes are specifically biological catalysts. So these are incredibly important in biological systems. Inside of you, there are a lot of key chemical reactions that ordinarily take place too slowly for the products to be formed in a timely fashion. Without these products, cells wouldn't be able to thrive. They wouldn't live. So enzymes evolved, and they help to speed up these reactions so the products that are needed are available at the correct time. A fourth function are the components of your immune system. The fancy name for this are immunoglobulins, the antibodies that will attack foreign invaders that are able to get through your skin into your system. The antibodies will seek out those foreign bodies and take care of them, get rid of them. A fifth function is the formation of hormones. We talked the other day about steroid hormones like testosterone and progesterone, but many hormones are actually proteins in structure and characteristics. Next year in biology, you will study and discuss hormones at great length. Later on, if you take human anatomy and physiology, hormones would be central to a lot of your discussions. And finally, proteins can be used for energy. Again, we saw this a bit in the laboratory you did involving energy content of food, where one of the components that could be consumed as combustion took place would be proteins. I referenced at that time when you see pictures in your history book of survivors of death camps, concentration camps, their bodies are wasted away. They were slowly being starved to death, and their bodies turned to the muscle mass inside of their own bodies and started to digest them, trying to utilize them for energy. Now let's talk a little bit about these amino acids. Here you see on this slide the basic structure of an amino acid. There really are 4 parts that are connected to a central carbon. Some of these are going to be familiar. You have a carboxyl group, carbonyl with a hydroxyl group. At the other end, you have an amino group. We talked about amines before when we talked about acids and bases. A hydrogen. And then that 4th position, again, you see that generic R, some kind of side chain. And it's this side chain that is going to characterize each amino acid. Here you see the simplest of amino acids, glycine. Again, you can find the carboxyl group, the amino group, the hydrogen, and in this case, the side chain is another hydrogen atom, the simplest of all amino acids. The character of that side chain determines how we categorize the character of the entire amino acid. And the reason is these side chains are the groups that are going to stick out from the side of the linked amino acids, and these can interact in the environment with different things that are in the environment. And when these chains get really long, what's going to be in the environment are other side chains of nearby amino acids. Let's now look at a whole set of amino acids. And on this slide you see what are categorized as polar amino acids. So as you go through and you peruse and you examine these amino acids, you will see groups that we typically have run into that we have identified as being polar. You'll see hydroxyl groups, you'll see amine groups, carboxyl, carbonyl groups, all of which we know involve atoms that aren't sharing electrons equally. They've become polarized, and that's why these amino acids with these side chains are classified as polar. On the other hand, here are a handful where the side chain involves atoms connected with pure covalent bonds. There's an even sharing, an equal sharing of electrons, and therefore they are nonpolar and therefore are nonpolar amino acids. Again, these side chains and their character is going to play a big impact as these proteins form and we link more and more amino acids together, forming larger and larger chains. Then you have flexibility in those chains to allow side chains, the side groups, to interact with each other. That's going to be really important throughout our discussion of proteins right now and your discussion of proteins next year in biology. Now, what do we do to link together these amino acids? Well, first of all, realize that when amino acids are being joined together, when molecules are being formed within the cell, there has to be a blueprint. We have to have directions that guide our cells in producing the right compounds needed for a whole variety of functions. So when these proteins, again, performing those 5 or 6 different functions we listed at the beginning of this discussion, you need to have a blueprint. When I want to make a hormone, I've got to have a blueprint, a plan that tells me which amino acids I need, when to hook them together, and therefore will guide the larger structure and the larger function of the resulting peptide or protein. So that's all going to reside in your DNA, which obviously is inside of the nucleus of a cell. That's where all of the plans reside that are needed to make everything in your body. And every cell, for the most part, there are a few exceptions, but virtually every cell in your body has the full blueprint of everything that would need to be manufactured. That's in the DNA, which really reside in what you know as chromosomes. So when it's time to make a particular protein, those chromosomes, these are enormous molecules, these DNA molecules, they have to unwind, and there's a mechanism to read the blueprint. And then we use what is called RNA, which will carry that information, that will carry that blueprint out into the cell, out into the cytoplasm of the cell, where the manufacturing is actually going to take place, specifically in an area known as the ribosomes. So the blueprint gets copied. We're not going to send the DNA out into the cytoplasm. We're going to send a copy of the information, and that's called messenger RNA. And that'll go to the factory, so-called factory called the ribosomes, and we're going to start reading, transcribing the blueprint. And there'll be a code, and the code will tell your cell which amino acid do we need to put in the first position on a chain. We have what's called transfer tRNA, whose job that is to bring in the correct amino acid. So that's where this would be happening. On the ribosomes, you've got the messenger RNA, you've got transfer RNA, you've got ribosomal RNA, you've got these different kinds of RNA. They're all involved in taking the information, translating it, and building the molecules. So you can see in this slide that an amino acid has been retrieved, and a second one has also been retrieved, and we're going to bring them together. And once again, just like you saw with the monosaccharides, there's going to be a special enzyme whose job is to connect these two amino acids to each other. And you can see the hydroxyl group on the carboxyl end of one amino acid is going to react with the hydrogen on an amine group on the next amino acid. We're going to pull a water molecule out. So we're going to dehydrate this, and the carbon will have lost a bond, the nitrogen will have lost a bond. They've got to get that bond back. How do they do that? They connect with each other, and this forms what's called a peptide bond. So the process is the same as when we formed a glycosidic bond, but now we're connecting two amino acids. We refer to this as a peptide bond. And just what you would probably think, if I want to digest a protein, I'd have to break that peptide bond. I'd have a digestive enzyme that would go back into that bond and add a water molecule, rehydrate it, and break apart the amino acids. Here you see a chain where we've got many, a couple dozen amino acids linked together. You can see I'm not showing the structure. I'm simply using the three-letter abbreviation for the amino acids. And you kind of get a sense even with this structure here, and this is called a primary structure, that it's pretty flexible, even the primary level of organization. Primary structures simply mean which amino acids are linked and what order are they linked together. So it's not only which are present, but what order did we put them in. So if I take the same amino acids and I change the order, I'd have a different peptide or a different protein. And you see that here on this next slide, where I've got two peptides. These are pretty short chain of amino acids. The first one called oxytocin. You see the 9 amino acids, so it's a nona peptide. And it looks just like this other peptide called vasopressin, except for a substitution of amino acids in the 3rd and the 8th position. Now again, this might seem kind of trivial, except that oxytocin is a peptide that is produced mainly in pregnant females right when they're getting ready to deliver their baby. Oxytocin levels spike in the bloodstream of the woman who's about to deliver, and it actually helps to stimulate the muscles in the uterus so that those muscles contract and a baby gets born, gets pushed out of its mother's uterus. It's also used to help produce milk. So once that mother delivers her baby, she can nurse the baby by producing milk, and the baby gets its nutrition from the milk. Vasopressin, on the other hand, is produced in all of us, and it actually helps us control blood pressure. This actually is used to stimulate muscles that are in the walls of blood vessels, and when they contract, it makes the blood vessel narrower, which would make the blood pressure go up, or those muscles can relax and make the opening of the blood vessels larger, which would make blood pressure lower. So vasopressin is actually a peptide that stimulates those muscles in the blood vessels. Two vastly different functions, simply because their primary structures, what we call primary structures, are different. We substituted in different amino acids in 2 positions, and that changes the function. Now, so that's the primary structure. What is the sequence and which amino acids are present? The next level of organization is referred to as the secondary structure of proteins. So as you can see in this illustration, the chain got bigger and bigger and bigger. And in this particular protein, it got big enough where the chain can distort. You've seen this when I've built models in class and very easily started to manipulate them, and you saw even with a fairly small molecule, you can really start to configure the atoms a little differently without actually breaking any bonds. And that's what happens when the chain of amino acids gets long enough. Then there'll be a distortion in the chain, and that's when the protein can assume a secondary structure. And here's one example called an alpha helix. It's a spiral, okay? And you can see in the left-hand portion of this diagram, it looks like a ribbon. That's how they're depicting the chain of amino acids. And you can see how it forms that spiral, that what's called an alpha helix. In the second 2, they're starting to fill in actually all the atoms that make up the central part of the chain. That's what you see in the middle. And in the third panel, you see where they put the side groups in. And the dotted lines, I don't know if you notice, the dotted lines illustrate side groups that now are linking to each other. And so in this diagram, which I really like about this, is you can see those side chains then, once they link to each other, they kind of lock the spiral in place, so it won't unwind. You would have to break those bonds. Those dotted lines represent bonds between the side chains. You'd have to break them in order to loosen up the helix and let it unwind. The alpha helix looks a little bit like a spring. And so an alpha helix would be important in a protein where you want sort of a springy kind of consistency. That a little extra springiness is important for certain functions, and that's what you would see, for example, in a tendon that joins a muscle to bone. You want it to be able to be spring-like to absorb some of the energy. Or in hair, if you looked at the microscopic structure of hair follicles, you would see that the protein that makes up hair is actually got an alpha helix kind of arrangement. So the secondary structure was important for the functioning of the protein that it represents. Now that's one type of secondary structure, but it was determined by the right amino acids in the right order so that those side chains can link. If you put in the wrong amino acids in the wrong order, your alpha helix may not form. So again, that goes back to the blueprint that was back in the nucleus in the DNA. So if there's a mistake, you might not be able to form your protein, and the protein won't be able to do its job. If we go to this next slide, there is an alternative secondary structure known as a pleated sheet. This kind of looks like an accordion. You can see there are multiple chains. I like the middle panel really illustrates that well. The middle and the far right one give you the idea that you've got chains that are side by side and then they're cross-linked, but they form like a little accordion. So again, that gives you a certain characteristic, gives you a little flexibility and yet makes it strong. And that's what you would typically see in muscle fibers. So muscle tissue is made up of proteins that are able to form what's called a pleated sheet. So again, slightly different function, but again, going back to what was the primary structure, which amino acids, and in what order are they, so that you can get a pleated sheet. Here in this slide, you see them side by side, and you can see that an alpha helix is a single chain just spiraled together, and a pleated sheet is actually multiple chains side by side, and they're cross-linked. Again, to maintain these structures, you have to have these side chains. That's why you have to have the right amino acid. Otherwise, you won't have the right side chains that can interact. Here you see 3 or 4 different versions. On the top there in that pink, you can see we've got, looks like nonpolar and nonpolar coming together. These side chains, well, that's going to be your dispersion forces acting that we talked about before. Sometimes you can form hydrogen bonding. You see in the second part of that diagram, you can form a hydrogen bond between side chains that are of amino acids that are at a distance apart. They're not adjacent to each other. They're pretty far apart. And you can see how this would hold this spirally configuration in place. And the third one there you see are ionic interactions. And here another slide that illustrates the same sort of thing, whether those side chains form, here oxidation reduction, that allows the side chains to link and hold the secondary structure. Okay, now if we make the chains even bigger, and as they get bigger and bigger and bigger, they get more and more complex, they get more flexible, and then you can have some proteins, when they're really big, have not only a primary and a secondary structure, but now can also have a tertiary structure. And here it looks like a giant glob. So you had a secondary structure like those spirals, those alpha helices, and they can just kind of literally fold on top of each other, and they form what's called a globular structure. And it's very apt because it describes exactly what the thing looks like. And again, what holds it in these shapes are going to be side chains interacting, and you get, again, either dispersion forces, dipole-dipole, hydrogen bonds, the same things we've run into before. This one, it's very hard might be hard for you to tell, but there's actually a pocket in the very center of this protein. So this protein's job would be to form that pocket to hold stuff. And that might be it's going to carry things around. Remember we talked about transport. Some proteins are, their job is to transport things. It would have a globular structure, much like this, forming that little pocket, that place where the molecule can fit in. This happens like with hemoglobin in your blood, or myoglobin. They have pockets for oxygen being carried around. This slide kind of illustrates that. Here you see a protein that actually has some alpha helices and some pleated sheets. And the thing is folded. It looks like a giant ribbon that's all folded on itself, and it's gone into a tertiary structure. Same thing here. You see the spirals, and then it's the whole little ribbon is folded upon itself, and you get this tertiary structure. Proteins often will have these tertiary structures where what we call that pocket, we call it an active site. It's a place where the chemicals are going to reside when the enzyme speeds up the reaction. And it speeds it up because it brings molecules close together. And those little pockets are designed to fit only certain molecules. So the enzymes don't act on every molecule, only on ones that they're designed for. We call that the lock and key model. You may have read about that in your book, or you'll be talking about that at length in biology next year. If you break those bonds, though, between the side chains, this big glob is going to start to unfold and the pocket will disappear. That happens when proteins are denatured, denaturation. Energy can cause that, certain chemicals can cause that, and the protein would stop functioning because its active site disappears when the thing unfolds. Other times, a molecule could mimic what's called the substrate and get into that pocket. And if it gets into the pocket, then the thing that should have been in the pocket can't get in there anymore. That's called inhibition. And you actually have the active site being blocked. And ultimately, a protein could get digested. It could lose its function that way, where you actually start chopping off those amino acids and you start breaking those peptide bonds through some sort of enzyme that rehydrates the bonds. And finally, well, here first of all, before we move on, a couple other slides just to point out here. Here's myoglobin, has a tertiary structure. I love this picture because you can see the alpha helix, and then you can see basically the alpha helix is folded on itself. And in the center where you have that little disc, that's where oxygen would be carried. So myoglobin carries oxygen in muscle cells. This slide, great illustration of denaturation, where those side chains have been broken. So in the upper left-hand panel, you've got your intact tertiary structure. In the middle panel, those linkages were broken, so it unfolds, and now it doesn't function anymore. Okay, if you refold it, it would regain its function. So that's a protein being denatured and then it being reversed and going and becoming functional again. And finally, as we wrap this up, talking about proteins, you can actually take multiple tertiary structures and put them together and form quaternary structures. And here is hemoglobin. This slide and this one illustrate hemoglobin, where you can see it's made up of 4 different chains, so 4 different tertiary structures, and you can see there are locations, pockets, where oxygen would be carried. So hemoglobin's job, obviously, is to carry oxygen in your blood. Hemoglobin is an example of a protein with a quaternary structure, so this is a really, really huge molecule. You also have antibodies. So here are immunoglobulins. You can see they've color-coded it, and you've got, it looks like 6 different chains there, 6 different tertiary structures, these have to be able to act upon invading organisms and deal with them. So it kind of makes sense they would be very large. And my last slide illustrates, some of the things that we, levels of organization we've talked about. Primary, all proteins have a primary structure. If the protein gets a little longer, then it can fold or it can spiral, and then you can have a secondary structure. If you get one that's even longer, then they could then fold on themselves and have a tertiary structure. And then you can even have ones that are even bigger than that, where you can bring multiple tertiary structures together to form a quaternary structure. So not all proteins have every one of these levels of organization. They all will have a primary structure, but the determination whether you have secondary, tertiary, or quaternary depends on how big the molecule is, and that's determined again by the information in the blueprint in DNA. That's proteins, so make sure you read the book. Make sure you look at the illustrations. Go back over my handout. Hopefully this helped you out and you have a little bit better understanding of what proteins do. And the real details will come next year in biology.